Every time a muscle contracts to pull a heavy load, or a red blood cell rushes oxygen to a starving tissue, a specific class of biomolecules is doing the heavy lifting: proteins. Yet, despite the massive scale and complexity of these biological machines, they are all constructed from an incredibly small set of simple units. That fundamental unit is the amino acid. It is the absolute base material for all biological architecture. This is the core paradox of biochemistry. The human body requires near-infinite structural complexity to sustain life, but it builds that complexity using a tiny, standardized chemical alphabet. This diagram illustrates the standard alphabet. Every amino acid shares the exact same backbone, a central alpha carbon bonded to an amino group and a carboxyl group. The variation comes entirely from this attached R group side chain, giving each amino acid its distinct chemical identity. Under physiological conditions, these molecules act as zwitterions. A zwitterion maintains a net electrical charge of 0 across the entire molecule, even though it simultaneously houses a positively charged amino group and a negatively charged carboxyl group. That specific distribution of electrical charge and the unique shape of the R group dictate exactly how this single molecule will react, bond, and behave inside the chaotic environment of a living cell. This chart categorizes the 20 standard amino acids based on R group behavior. On the left, nonpolar side chains repel water, while polar side chains below attract it. On the right, electrically charged side chains divide into negatively charged acidic and positively charged basic groups. From a nutritional perspective, this table divides the 20 ingredients. The red column lists 9 essential amino acids. The human body cannot manufacture these and must acquire them through diet. The blue column lists non-essential ones the body synthesizes on its own. Every single hormone, every single enzyme, and structural fiber in your body depends completely on sourcing and precisely combining these 20 specific chemical ingredients. To build anything useful, these units must link together through a condensation reaction. When the carboxyl group of one amino acid meets the amino group of another, they shed a molecule of water. What remains is a strong covalent connection called a peptide bond. This illustration visualizes how that process scales. 2 linked units form a simple dipeptide, but as the process repeats, head to tail, over and over, it creates a massive long-chain polypeptide. These covalent peptide bonds act as the grammatical syntax of life. They turn individual chemical letters into long biological sentences, forming the backbone of proteins. A linear sequence of amino acids is called a primary structure, but a straight chain cannot do biological work. To become functional, it must fold. At the secondary level of structure, hydrogen bonds naturally pull the chain together. It either coils into tight alpha helices or folds into flat beta-pleated sheets. This 3D ribbon model represents the tertiary structure. As those varied R groups repel and attract one another, they force the entire molecule to crumple into a highly compact, complex, 3-dimensional shape. Some proteins require a quaternary structure. Here, multiple folded polypeptide subunits physically lock together to form a supercomplex, exactly like the hemoglobin in human blood. A protein's ultimate physical geometry is completely determined by that original, linear sequence of chemical ingredients. And that precise geometry is the only reason the protein can act as a catalyst, a structural support, or a chemical messenger. The resulting shapes dictate exact functions. Fibrous proteins, like collagen, form long, water-insoluble threads that provide tough connective tissue and structural support. Conversely, globular proteins fold tightly into water-soluble spheres, making them highly mobile enzymes and transport molecules. But these structures are vulnerable. When exposed to extreme heat or highly acidic pH levels, a protein undergoes denaturation. The weak bonds holding its 3D geometry fail, causing it to rapidly unravel back into a disorganized, useless string. Ultimately, biochemistry relies on precise physical fits. If a protein loses its architectural shape, it instantly loses its capacity to interact with other molecules, ceasing its ability to sustain life. Today, the pharmaceutical industry leverages this exact biochemical mechanics. By understanding how amino acid chains fold, laboratories can synthesize therapeutic proteins, from lab-created insulin to highly specific monoclonal antibodies. By engineering a structure matching the specific spatial requirements of a targeted cellular receptor, pharmacists can correct metabolic errors and treat disease with molecular precision. Fluency in this chemical language moves medicine beyond simple observation, providing the literal building blocks for the next generation of healthcare.